take-home message was that homeostasis is the body's relative position when you're healthy. In a clinic, we use a bunch of different things to determine whether somebody's in homeostasis. We do blood pressure, we do pulse, we do respiratory rates. We also do blood screens and do blood analysis, urine analysis. All of those tests are trying to determine whether or not you're in homeostasis. And when you do all those tests, there are boundaries, a low and a high, that if you're in homeostasis. And if you're outside of that, you're abnormal and you're not in homeostasis. So the whole goal is to keep your body within those boundaries which we have concluded established health. Right? And then what we talked about was that homeostasis is dynamic. It changes based upon daily activity. It also changes as you age and maturate from a baby to an elderly. So as I just said, the two systems that are involved are the nervous system and then the endocrine system. And because the nervous system works on weak electric currents that we're going to talk about when we do membranes, then that information travels through the body very, very rapidly. Okay. Hormones, which are chemicals that are released by one tissue to the blood and then circulate in the blood, are slower because the hormone has to be made, released to the blood, and circulated. Okay. So we kind of think of the nervous system as the rapid response to changes, and then hormones usually are a slower response to changes. And I said, as in a clinical setting, we measure a bunch of things to determine whether somebody's in homeostasis. So we can do blood carbon dioxide, blood oxygen, body temperature, blood pH, blood pressure. So we can do a bunch of different things to ascertain that. So one of the examples I could give you is that you are essentially a walking bag of water. And the beauty of the tissue we talked about yesterday, which is keratinized stratified squamous, is that it creates a impermeable barrier, so that essentially you are a pond walking around and your cells exist in an aqueous or water environment. So that's pretty amazing in itself that we manage that. But to do that, we have to manage where water is in our body. So if you just kind of look at the relative distribution in the body, women are about 55% fluid, men are about 60% fluid. So we are essentially largely water. And so that's good news when you get on the scale. Most of that's just water at 8 pounds per gallon, right? And what we do is we compartmentalize the water. So we compartmentalize most of our water inside of cells. So we have all these cells, like we started looking at yesterday, cuboid cells, columnar cells, squamous cells. And so the water that we find within a cell, largely helping make up the cytoplasm of the cell, we refer to as intracellular water. So intra is a prefix that usually means within. Okay. And the way we manage our water is we have a urinary system that is designed to help get rid of excess water, and we drink to replace water. Okay. So we use our digestive system and our urinary system to kind of manage the water balance in our body. So we have to use our cardiovascular system to transport water to where we need it. Okay. So within a blood vessel then, we have what we call blood plasma. And blood plasma is mostly water. Okay. So about 20% of our water is over here in the blood plasma area, and about 80% of our water is over here in our cells. And then we have to have a way in which this fluid can go from the blood plasma to the cell. So we have spaces between cells. So we have this space that exists between cells. And so we call that interstitial fluid, or IF. And it accounts for, this should be about 2/3, excuse me, 2/3 over here. And then these two represent 1/3. And the IF is actually 80% of the 1/3. Okay. So the take-home message when you look at this is that if your cells take on too much water, then they swell, 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 and burst. If you put too much water in the plasma component, then blood pressure goes up. So what we have to do, what our body does on a daily basis, is manage water and manage where it's found in your body to maintain homeostasis. So there's actually a woman in California who responded to a radio station contest. And it was when one of the new gadgets that you play games with. And the contest was the person who could drink the most water in the shortest period of time would win. And if I recall right, she drank about 5 gallons in 20 minutes. And when she left, she complained of a really bad headache. An EMT called and said, "Don't let her go, call the ambulance, get her." She died. Because all the water she ingested went into the plasma. And because there was so much water in the plasma, a lot of it moved out into interstitial spaces. And as she loaded her interstitial spaces, the water moved into cells, and neurons in her brain actually were rupturing and exploding. So the kidney couldn't keep up with that kind of input of water. So this is just a classic example of homeostasis we deal with on a daily basis. So to do homeostasis, your body really has to know what's going on. So our body's loaded with receptors that send signals to the brain. And so the first thing we have to have is some type of receptor that's monitoring something that's going on in your body. We have a bunch of pressure receptors that we depend upon that help us regulate body fluids and blood pressure. Okay? And then we have to have a control center. So that's the key to our central nervous system, our brain, our spinal cord, is all these impulses that are created by these receptors have to be sent to the brain, and then the brain has to interpret what's going on. All right, and then what happens then is the brain then sends a signal back to the body to adjust some function within the body. So that aspect is an effector. So the way to think about it is the core of this idea is that we have a central nervous system, a brain, and a spinal cord, so the CNS. And all over our body we have receptors that are responding to changes. They send signals in, and then the central nervous system returns it and sends a signal back to something like, that would be an effector that would make some change, okay, as to what we're doing. And so we're dependent upon this throughout our lives, that this communication system, sending signals in, appropriate interpretation, and sending signals back to make adjustments is actually going on. Okay. So a good example, and then there was actually a term on the first lab that I actually forgot to cover. So this gives me an excellent time to catch up on it. So at the bottom of the first lab there was the term mediastinum or mediastium. And so the way to think about it is that is the area between your lungs in the thoracic cavity. It would contain your heart, it would contain all the major vessels coming to and from your heart, your trachea, your esophagus, depending on what level we were looking at. So the black dashed line here is actually outlining the mediastinum. So you can see it's starting with the body of the vertebrae and going all the way to the front, encapsulating everything between your lungs. So So I just forgot to cover that. So one thing we have to do is we have to adjust blood pressure. Because blood pressure changes. If you had to have a situation where it was critical, you had your lowest possible blood pressure, when would be a good time to take it? Before you get out of bed. Why do you think in the hospital they come in and it's like, why are you waking me up at 5 o'clock in the morning to do this stuff? Because that will be your lowest blood pressure. As soon as your feet hit the floor, blood pressure goes up. Okay. So what we have is we have a way in which blood pressure can change depending on what we're doing. Standing, walking, laying, climbing stairs, riding a bike, that all changes blood pressure. So your brain has to know what's going on all the time with blood pressure. So we have these pressure receptors in our major arteries. So we have them in our aorta, we have them in our carotids and our neck. And what they do, they're called baroreceptors, and they send signals to the brain about the relative amount of blood pressure. So the brain is really self-serving in this regard, because what we know from history is that before we knew how to manage blood pressure with drugs, that people were dying at a higher percentage of strokes much earlier in their life than they do today. So as blood pressure goes up, you put stress on the walls of blood vessels, and the ones in the brain seem to be very susceptible to damage to the walls with chronic long-term blood pressure. So what happens is we want to be able to control blood pressure. So what we have is we have a receptor, which is the baroreceptor. We have the brain, which is our control center. And then what the brain does, and when we get to the nervous system, we'll talk about the specific areas of the brain that do it. It sends signals back to the heart saying, hey, stop beating as hard, and tries to bring blood pressure down. So the interesting thing is that for most of your life, your body does this pretty darn well. But when you start reaching your late 40s and early 50s, it stops doing it so well. And so people used to enter high blood pressure events in their late 40s, early 50s. And if you went back to the 1920s, the average age of death of men was 58. And now it's in the 70s. And it's largely due to two drugs, blood pressure and control of lipids. Okay. So it's pretty cool. All right, so there are two ways that we can do this idea of having this feedback mechanism where we have a receptor, send signals are interpreted, and the effector comes back. And what we do is we divide them all into positive feedback mechanisms and negative feedback mechanisms. So the first take-home message is most of the body works on negative feedback mechanisms. They're much easier to do. What you're trying to do is keep some activity of your body at a low level, and if it accelerates, your goal is to get it back down to that low level. Positive feedback mechanisms would be where your body is at a low level and you keep trying to accelerate it all the time. So it's an opposite thing. So it's much harder to control something by accelerating it all the time than it is to try to keep it at a low level. So positive feedback changes, a good example would be childbirth, and it's hormone driven with a hormone called oxytocin. The more oxytocin you have, the more uterine contractions you have, and the harder the contractions are, and the longer the contractions are. And it just keeps accelerating until the baby's out. And then it goes back to normal. Blood clotting would be another example. So, because water balance is so critical for us, if you have a hemorrhage, you don't want that to keep bleeding. So there's this cascade of biochemical reactions that occur with clotting factors that accelerate to create a clot. Okay. And then a couple others would be like protein digestion. So the first example, childbirth, is actually, again, driven by a hormone. So long story short, it's actually baby that decides baby wants to come out. And baby sends a signal to mom's brain, which is a hormone that goes from baby's placenta into mom. And then that triggers the baby to start dropping, and then that stretches the cervix. And then as the cervix stretches, the stretch receptors in the cervix send a signal to the brain that it's being stretched. And then the brain releases a hormone called oxytocin. And then oxytocin is a strong, smooth muscle contractant, and it makes the uterus contract harder and harder and harder and harder until the baby comes out. And then after that, then you go back to normal activity. So it's just a classic example of a positive feedback mechanism. And actually nursing or lactating is as well. So when baby is nursed, the stimulus of the nipple triggers oxytocin release, and the oxytocin triggers smooth muscle in the milk ducts, and that moves milk quicker to the nipple. So some women that are breastfeeding, when the baby gets a little older, third or fourth, fifth, sixth month, where they're still nursing, they'll go out to eat and leave the baby with the babysitter, and then a baby will cry, and they'll get that oxytocin release and milk let down. So they actually have to go prepared because they're going to have a wet shirt. So it's not only stimulation of the nipple, but actually auditory stimulation as well. All right. Yes.
My last time teacher told me this, and I just see if I was actually sure that oxytocin is the drug that causes a very big connection between the mother and the child while
Yes.
nursing. It's basically your nursing lockdown.
Yes. And so he's asking, oxytocin apparently impacts emotional areas of the brain significantly. And so a lot of women go through what's called a postpartum depression after the delivery. And what we've been able to do is see that women who don't have these peaks of oxytocin through lactation sometimes go into much deeper states of depression than women who don't. Now, there's some recent studies that actually show, so this hormones are something that we've known about, but they're so hard to study, and there's so many intricate factors affecting them that our technology is leading us to understand them at a level at which we never understood them before. So 10 years ago, I would have never said that men have oxytocin circulating in them, but we do. And women do that aren't pregnant as well. And it seems that oxytocin may and will be a very important drug to brain function outside of what we've thought, what we've understood all the time. So even more than what he told you, it may play a bigger role than we've ever known. Recent studies have looked at circulating levels of oxytocin in men and women and compared them with depression and some other events. Right. So the idea that I've been trying to get you to think about is that as long as we're in homeostasis, then we're happy and healthy. It's as we get out of homeostasis that we need clinical intervention. And so what we do is we take those things that we measure to determine whether somebody is in homeostasis, and then if you're out of it, then we categorize the reason why you're out of it. So a classic example would be blood sugar. If you go to the doctor and you get a blood screen, one of the common things they do is screen blood sugar. And there's a range where you're abnormal blood sugar. Well, if you go to the doctor and all of a sudden your blood sugar is skyrocketing, then they have to figure out why in the world your blood sugar is skyrocketing. Well, one of the reasons could be is that your pancreas has stopped producing insulin, which is required to manage blood sugar. So if your blood sugar is really high, and we have looked at your insulin levels in your blood, and that's the issue, then we could categorize you as having a disorder. And the disorder would be diabetes, okay? So what we do is we take all those signs, we do an analysis of how well you're doing. If things are out of stray, then we have to figure out why. And what we've done is create a bunch of disorders and diseases that explain those symptoms. So the art of the clinical environment is understanding the symptoms of diseases and disorders. So a disorder is a general term for any change or abnormality of function in the body. Disease is a little more specific for an illness characterized by a specific set of signs and symptoms. Right? So a local disease is one that that's limited to part of the body. A systematic disease is one that affects the entire body, like diabetes would. So what we have to do in a clinical environment then is to collect data and use that data to tell us where we're going. Okay. So as we said, a disease is a specific term. So signs are objective changes that a clinician can observe and measure, i.e. that's all those tests that we just talked about. Blood pressure, pulse, respiratory rate. When they listen to your back and say, breathe in, breathe out, those are all signs. They're collecting signs that might tell them something's wrong. The other part of going to a physician is they make you fill out that little chart and have you had this, have you had that, have you had this, have you had that. What that is what those aren't signs, those are symptoms. And those are subjective changes that have occurred. So I can't look at any one of you and know whether you had a headache. How do I know whether you had a headache? You have to tell me. There's the biggest dilemma with healthcare, is many people are not forthright with what's going on. And elderly people in particular will not tell a lot of things going on because they're either embarrassed or their mind themselves don't want to accept what's going on. So that makes it really hard to do diagnosis because you only have part of the picture and you have to guess at the other part of the picture. So that's something you always have to keep in mind. So we get signs by doing a physical exam. We get symptoms by doing what we would call in a clinical environment, a history. Right? So it's something to keep in mind as you go forward. So as my mother aged, I take her to the doctor. He'd ask her questions, and it always annoyed me because she would say something and it's like totally a lie. And either I have to say, no, she's wrong right in front of the doctor, and I kind of wish he would say, hey, can we go outside and talk? And I'd always have to go, no, that's not quite true. And then my mom would look mad. Because otherwise the doctor wouldn't know what was going on. All right, so that's the end of lecture 1. So lecture 2 is actually got a lot of stuff I expect you to know from general biology. So I actually created the lecture to guide you in a review of stuff you should know from general biology. So some of it I'm just going to gloss over and say, review this please. This is what I expected. There's some stuff I really want to talk about because I want to make sure you have a good understanding of it. Okay. So just as a quick history, because I always want my students to go away, if they've learned no anatomy, at least knowing how science works. And the fact that science will change. And just because we have to change what we know in science doesn't mean we were wrong the first time. We just didn't have as clear a picture as we do now. That makes sense. So Hooke was the first one that named the cell in the 1600s because he created a crude microscope. And what he could see is living tissue actually was made up of this repeated entity, which he called a cell. Now, he was probably either fortuitous or just lucky that he chose plant tissue to look at because they have cell walls that define the boundary of cells. If he started with animal tissue, we probably wouldn't have got there quite as fast as he did. So it's just kind of fortuitous. Pasteur in the 1800s was the one that did an experiment to show that non-living entities didn't convert to living entities. Now, to you guys, that might sound ludicrous, but before Pasteur, people believed that a rock could turn into an animal. And that may sound totally ludicrous to you guys, but you're thinking about, your knowledge base is so much different because of all we know about science. Yeah. So he was the first one that said that, wait a minute, you know, water doesn't get contaminated by bacteria simply because little things turn into bacteria. He said that actually it has to get there from another mechanism. So what he did is he took some containers, closed some, left some open, and the closed containers never got life, and the open was in. It was like, okay, so this water is not turning into something. Okay. And then, based upon a bunch of other research, the modern cell theory emerged. And so what the modern cell theory tells us is that all organisms are composed of cells and or cell products. Okay. The cell is the simplest structural and functional unit of life, although viruses are beginning to test that, because we have viruses like the HIV virus that mutates all the time, it changes. So they're beginning to test that piece of it. Organisms, tissues, their organ structure and function are due to the activity of cells. That's what we began to work on yesterday. Why do we have squamous cells in certain areas of our body? Why do we have cuboid cells in certain areas of our body? And why do we have columnar cells in certain areas of our body? And that's because those organs have specific functions, and the cell shape is driven by cell function. So that part we've actually begun to play with, is that these organs are based, their structure and function are interconnected to each other. So cells only come from pre-existing cells. That's what we know right now, but it doesn't answer where the first cell came from. I don't know that we could ever do an experiment to establish that, so it's kind of out of the realm of science anyway. So cells of all species have many fundamental similarities. So I know you talked about this, but you probably didn't think about it in this context. Every organism on Earth, bacteria, plants, fungi, and animals all use ATP as an energy molecule. That means that every organism on Earth shares common genes for the construction of ATP, the enzymes that make it. So when we look at genetics, the similarities are amazing, because we have very similar genes across broad spectrums of species. So it's really kind of cool to think about. Okay, so human cells are really pretty darn small. The largest human cell is the egg that you arose from. A human egg is visible with the naked eye. You don't actually need a microscope to see a human egg. Okay. So other cells are amazing. So neurons in your brain have to expand distances to communicate. So a neuron that controls the movement of my leg has its cell body where the nucleus is in the frontal lobe of my brain, and then it has an axon that goes through my brain down to my spinal cord and exits somewhere at the base of my spinal cord. So that neuron is that long, one cell. And then a new neuron arises in the spinal cord and carries the information down to my foot. So you have neurons that are as long as your legs. So what we're going to do is we're going to look at a bunch of cool cells over time and get away from the idea that a cell is a little round thing that has a nucleus, and they have all kinds of cool shapes for function. Right? So the one dilemma that we began to talk about yesterday with red blood cells and talking about surface to volume ratios was that what limits the size of cells is that when cells grow, volume increases faster than surface area. So when we think about a cell, and I've got actually the next one does a good job of that, and it impacts the ability of the cell to take things on. So this is kind of nice because cubes are easy to work with. It's easy to calculate volume of the cube, and it's easy to calculate surface area of the cube. So if we looked at cuboid cells like we were looking at yesterday, simple cuboid cells, then the average cuboid cell that you looked at yesterday was probably around 10 microns when we were looking at the kidney and stuff, right? And they're cubes, so they're 10 microns by 10 microns by 10 microns. So the diameter of the cell is essentially 10 microns. The surface area then would be 10 times 10 because each surface is 10 microns high and 10 microns wide. And a cube has 6 surfaces. So what you would do is take 10 times 10 times 6 to account for all that surface area. Okay? The volume is actually looking three-dimensionally at it. And so not only are we looking at height and width, but we're looking at depth. So now we have to look at 3 different measurements. So then it's height, width, and depth, 10 times 10 times 10. And then the 10s are multiplied. So what we end up with is surface area as a square and volume as a cube. That's why volume increases faster than surface area, because it's a cube, all right? It's to the 3rd power instead of the 2nd power. So if we just if we double the diameter of the cell, and we make it 20 microns, then the surface area becomes 2,400, but the volume becomes 8,000 or 8 times larger than it was, so it's increasing at a much larger rate. So you can actually model this with a computer and actually put it in and figure out how big the largest cell could be. Now what's fascinating is one of the largest cells we could observe is an ostrich egg. It's one cell. That chicken egg you eat for breakfast, if the chicken never met a rooster, is one cell. You're scrambling one cell for.
Would a nucleus increase in size along with the cell, or would it just make it?
No. But the biggest dilemma of what we're talking about is if you have a cell like this, it's easy to get things to the center of the cell. If you have a cell like this, it's much harder to get things to the center of the cell. And that's the dilemma. It's hard to provide oxygen and stuff and other nutrients be inside the cell. Yeah.
So would the yolk be in the cell or the whole egg?
The whole egg.
The whole egg.
Yeah. So the way to think about it is that eggs have a membrane that helps define their boundary. So if you hard boil an egg and you crack the shell, there's that little membrane on the inside of the shell. It's the boundary of the cell. It's not the cell membrane, it's actually a membrane that different, we call it an extra embryonic membrane. All right, so one thing we began to play with yesterday is cells have different shapes. And what I tried to get you to think about is that we started largely as a mass of polygonal cells. And what happens is a process of differentiation where polygonal cells then convert to different shapes of cells as they maturate. And then we see that same thing when we do mitosis, as we do mitosis with polygonal cells. And then those cells have to change shape. So we could see that yesterday when we were looking at stratified squamous, where the underlying cells were more polygonal, but as it got pushed to the surface, they became more and more flat cells and squamous cells. Okay. So what we're going to do is we're going to kind of look at cells as we go forward. So we've already looked at squamous cells, cuboid cells, and columnar cells. And those all belong to what tissue group? Epithelium, yeah. Epithelial cells. Yesterday when we looked at red blood cells, which would be the discoid cell over here, we also saw that we had these kind of big balloon cells that had nuclei, and those were white blood cells. So white blood cells are an example of a spheroid cell. Red blood cells are an example of a discoid cell. The cell that I was just talking about, the neuron, that is really long, is actually a stellate cell. And then one of the groups we looked at yesterday was actually this guy right here. It's a smooth muscle cell. So they're fusiform or spindle-shaped cells. And as we go forward and look at skeletal muscle cells, then skeletal cardiac muscle belong to a group we call fibrous cells. And so what we're going to do is we've actually looked at a lot of these cell types already, and we're going to look at some more as we go forward. And the take-home message is all of the cells up here started their lives originally as a polygonal cell and then differentiated. So this is the part where I'm going to depend upon you to do a little review. The way cells function is they have internal structures that are like organs, but since they're small and inside a cell, we call them organelles. So what I want you to be able to do is recognize an organelle, know what its function is. And if we know function, then the thing I want to stress to you is that not all cells have the same organelle. Some cells have many of one type of organelle, but other cells don't. Because what drives function of a cell is the amount of a particular organelle. So we were just talking about the endocrine system as a system that controls homeostasis, and it controls it by producing hormones. So if I take hormones and look at them chemically, I can put all hormones into basically two groups of hormones. Non-water soluble hormones that are all based upon a molecule cholesterol that they're constructed from. And then water-based hormones, of which many are proteins. So if you just look at rough ER and smooth ER, then smooth ER makes, remember, lipids. So then our cells that produce steroid hormones are going to be well endowed in smooth ER. Now the other group were proteins. So where are proteins made? So ribosomes make proteins, but usually within cells we find free ribosomes kind of hanging out in the cytoplasm. And we find ribosomes associated with ER, and then we call that rough ER. Well, free ribosomes appear to primarily make proteins for use inside of the cell, and rough ER makes proteins that are going to be exported. So a cell that's making a protein hormone is going to have lots of rough ER. Voila. So when we look at cells, they have different characteristics of these organelles. We talked about sperm yesterday. Sperm have none of most of that because their goal is to carry a nucleus to the egg. So they have lots of mitochondria to make flagellar action, but you would find very, very little ER in them at all. So the cells specialize in function. Organelles have to follow suit. All right. So, and there's a great picture with a chart in your book that does all that for you in terms of what organelles do. So the one I want to look at in detail is the membrane, because that's what really defines the boundary of a cell. And that's what allows things to enter and exit a cell. So the membrane becomes critically important to a cell. So there was a guy that did a clever experiment. And again, it's one of those things where he had a little insight or he was just darn lucky when he did his experiment. So what he was doing was using red blood cells. And I said lucky because they don't have a nucleus, so they have no internal membrane based on the nucleus. And they have very little rough ER and smooth ER because they're bags of hemoglobin. So he was fortuitous in that there was not much internal membrane material in the cell itself. And what he did is he knew the average surface area and volume of a red blood cell. And so what he did is he put red blood cells in distilled water, when you put a red blood cell in distilled water, they swell and break. All the hemoglobin, because it's heavy, drops to the bottom of the beaker. All of the lipids from the membrane, because they're non-polar, float to the surface. He was then able to calculate the surface area of fat that he had on the surface of the water, and it was twice what the surface area of the cell was. That's how we figured out that membranes are bilipid layers. Because we had to figure out, okay, how come I have twice the lipids I thought I should have? Because they're bilipid layers. And then if you just understand water and polar and non-polar, because they're phospholipids, and this is charged and this is non-charged, if you took a bunch of phospholipids in a chemistry lab and you stuck them on a beaker of water and you shook the beaker of water all up so they intersperse, the phospholipids will automatically form bilipid layers because the charged entity will orient toward the water molecules and the non-polar molecules will come together. Yeah. So it's really easy to make a bilipid membrane. Just take fat, phospholipids, shake it up, they'll form little spheres because of the way water interacts. Remember I told you water was, because it was polar covalent, it was one of the most critical things to life on Earth. Okay, so as we go through the class, we're going to look at different entities found within these membranes. So there are bunches of examples back there: channels, pores, glycoproteins. So I like this chart because it just kind of puts them into categories. So channels are like windows in a membrane. And without a channel, an ion might not move very well. But if we have a channel, an ion will move through the membrane readily. So we actually, and they're proteins. So our cells actually can put more channels in, take more channels out of a membrane to control what can enter the cell. So we're going to talk about channel proteins because they're fundamentally important to the way your nervous system works. It's cardiac muscle, smooth muscle, skeletal muscle. None of that would work without these channels. Okay. So the other thing is that we're going to review this is that we have kind of two ways in which things can move through cells. Passively, where we don't have to expend any energy, and active transport, which requires us to expend energy. And active transport always requires unique protein molecules that help move things through cells. And so those are called transporters, because they actually help transport things through cells. So we're going to look at different types of transporter proteins that we find in the cell as well. So the way hormones work is that when a cell is exposed to a hormone, it has to have a unique protein that the hormone can bind with. And then once the hormone binds with this protein, then it elicits a reaction by the cell. So things that are embedded in membranes or internal in the cell, and which another molecule has to bind with, are called receptors. So we have these receptor proteins. So if you guys, most general biology folks talk about the Krebs cycle, it's probably your favorite part of the class. Cellular respiration. But my point, the only point I'm trying to make here is, remember on the mitochondria, there were enfolded membranes inside the mitochondria, and that's where the enzymes were kept organized, were on those membranes. So not only within mitochondria do we find enzymes embedded in membranes, but on cells themselves as well. So we can have enzymes that are embedded in membranes as well. So does anybody know their blood type? We'll figure that one out next quarter for you, because we actually do blood typing in the lab. Well, the reason why we can do blood typing is because there are certain cell identifiers that are in membranes. And so if you have blood type A, it's because you have a unique cell identifier that we call A because it's short for something. So that would be blood type A. If you have blood type B, it's because you have a different cell identifier. If you have AB blood, so this would be A type blood, B type blood. If you have AB blood, you actually have both of those cell identifiers. But the amazing thing about the world around us is the most common blood type is O. And if you have O, you have no cell identifier. That's why O is the universal donor, because it's an unidentified cell.
So what's the difference between positive and negative?
That's a different identifier. Okay? So not only can you have A, B, and O, but you have what's called RH factor. So you have RH factor. And if you have the RH antigen, then you're Rh positive. And if you lack the Rh antigen, then you're Rh negative. And the most common blood of all blood types is positive, having that antigen. So there are some really cool things that have been done, particularly since we've been able to sequence genes, is in looking at blood types, is that we're now to the point where we know that that as we looked at human history, that probably we started with a particular blood type that was either A or B. And we know that there are mutations of each other, but we don't know which one was first and which one was the mutation. O-type blood is a mutation of A and B. And it's a phase shift mutation that creates a stop clause so that you don't produce the cell identifier because the gene's mutated to produce the cell identifier. Yeah. So what we do know is O is probably a secondary blood type that arose from a gene mutations. And it's the most common. And it's the most common because it mysteriously has genes, seems to be combined with genes that prevent you from getting certain diseases. That would be natural selection. Yeah. So if you have O-type blood, you're less susceptible to malaria. Well, the human civilization, however, started in the tropics. We weren't running around in the Arctic without clothes. So because it started in the tropics and malaria is a tropical disease, then if you had a capacity to survive malaria better than someone else, then you left more kids. That's natural selection. All right, so the way we can create an electric potential in our body, so this is kind of cool. We use a machine called an electrocephalogram, and we can measure brain waves. And then we can tell whether somebody has had brain damage by the change in brain waves. If you have no brain waves, we consider you brain dead. Okay, those are all electric currents. You can put an electrocephalogram apparatus on someone's head. Instead of connecting it to the machine that is graphing the waves, you can amp it and connect it to a toy train. And then the train will go around, and it's your brain that's driving the train. And you can actually do that with someone and ask them questions. And the harder the question and the more work they have to do to retrieve and answer, the faster the train goes. So our brain works on, we work on electric currents. That's why when you stick your finger in the socket, everything gets goofed up because some other electric current just overtook all your electric currents that were controlling your body. To do that, we have to keep ions in different concentrations across the membrane. So we have to create what are called concentration gradients. So if you look at the upper one, what ion, and so both of these, on top of the membrane is interstitial fluid. On the inside of the membrane, on the bottom of the membrane, is cytoplasm, okay? So we're looking at the cell relative to the pond it lives in inside your body. All right? So what ion is more common in interstitial fluid up there? The green ball, what's the green ball? So what positive ion is more common in the cytoplasm, which would be below the membrane? Potassium. So what we do is we maintain a concentration gradient where there's more potassium in our interstitial compartment and more sodium in our interstitial compartment and more potassium in our cytoplasmic or intracellular compartment, right? If we can maintain a concentration gradient, we can create a charge disparity on the membrane so that the outside of the membrane has a different charge than the inside of the membrane. And once we can create a charge disparity over the membrane, we can create an electric potential. So our brain sends signals all over our body by creating these concentration gradients that create these electric potentials that create electric impulses that we can drive a toy train with. So that's pretty cool, isn't it? All right, so how do things get inside and outside of cells? So the easiest way, which requires no energy on your behalf, is diffusion. So how important is diffusion to your body? No diffusion, dead. How do we get oxygen across the surfaces of our lungs? Diffusion. That's why we have squamous cells. How do we get carbon dioxide out of our blood into our lungs? Diffusion. No diffusion? No exchange. You're in trouble. And we use it all day in our everyday in our lives. We just take it for granted. So many people put perfume or cologne or something on. Voila! Why did you do that? Because you got it concentrated on your skin, and it diffuses all day off into someone's nose. And then you smell good today. And then after a while, diffusion occurs, and you don't smell as well because most of it's already diffused off your skin. So then you have to reapply it if you want to smell. Isn't that just amazing? Simple diffusion in action. All right. So what factors affect diffusion? Temperature. So what's temperature measured? It's a physics question.
Quickness of particles. The quickness of the particles moving?
Yeah, movement of particles, exactly. So, and you said quickness, so are particles moving faster when it's cold or hot? When it's hot, okay. So this is the part of science I just love. So if you're in a clinical environment and you stick a thermometer in someone's mouth and you say, oh, they're 98.6, you just measured how fast their molecules were moving. So why do we feel like crap when our temperature drops way down, like hypothermic events? Because molecules are not moving as fast. Why do you feel like crap when your temperature gets to 106, 107, 108? You're probably going to die.
Because your proteins don't work.
Because now your molecules are going like crazy, and you're changing the shape of proteins. Yeah. Wow. So our body is really designed to only like a narrow range of molecular motion. Why? Because it drives diffusion. So if your core temperature is 92 around in your heart and your lungs, you cannot drive oxygen into your blood very well because the rate of diffusion is really low. You get really lethargic, really tired, want to go to sleep because your brain is starving for oxygen. Yes. Simple diffusion. Yeah. So temperature, if we increase molecular motion, we would increase the rate of diffusion to an extent. There's there are bad things about temperature. So if you were at the center of a nuclear bomb blast, you would completely diffuse apart because your molecules would move so fast that you would just vaporize.
Is that still a factor on cold-blooded animals?
That's why you see snakes laying in the sun. God, please warm up. But they're much more tolerant to changes in oxygen because they are cold-blooded. So their body's used to not having the same metabolic rate, where we have the same metabolic rate. So that drives us to have this oxygen availability all the time. Okay. So what about molecular weight? Bigger the molecule, the faster the movement? No, slower the molecule. So hydrogen would probably be the fastest molecule to diffuse, right? Yeah. So let's look at two, carbon dioxide and oxygen. O2, CO2, which is a bigger molecule? Yeah, because these two are identical. You add a carbon to it, it makes it bigger. So intuitively, which one should move in and out of your lungs the easiest? O2, and that's exactly true. So steepness of the concentration gradient, that just makes sense. That's a perfume thing. Put the perfume on. So while there's a lot of perfume on your skin, you smell much better because it's diffusing at a faster rate. As the concentration gradients drop, diffusion rates drop. Okay. Membrane surface area. Highest surface to volume ratios are critically important, particularly in areas in your lungs and stuff where diffusion occurs. We have squamous cells because squamous cells have a very high surface area to volume. Red blood cells are biconcave because they have a higher surface area to volume, which makes them better for doing diffusion. Yes. And then membrane permeability. Now, how can we change membrane permeability? Add channels. And then you're going to change membrane permeability. Cool. Okay, so what we've been talking about is diffusion. And the problem with diffusion is it always requires a concentration gradient. Well, you can't always develop a concentration gradient. So if you can't do it, and so if we had a channel to increase the rate of diffusion, then we're doing facilitated diffusion. Does that make sense? So we can think of simple diffusion as something being able to move through a membrane. If it can't move very well, we have to add a channel protein to the membrane that helps it move. So then we're facilitating the diffusion by adding something to the membrane. And then the other, it would be active transport. The thing about active transport is it requires ATP or energy. So when we look at active transport, there are uniporters where one thing can move, and our body has them. But what you're going to see as we talk about this and go forward is that our body is really dependent on what we call coupled transport or co-transport. And we have two mechanisms for doing it. If we move two things in the same direction across the membrane, we call it a symporter. And if we move one thing from inside the cell to outside the cell while we're moving something from outside the cell to inside the cell, then it's an antiporter. So I do want you to know what a symporter is. Two things move in the same direction. And an antiporter, two things move in opposite directions. Because they're going to be critically important to our discussions in the future. All right. So I showed you that one picture, and what we saw was that if we looked at the membrane, we had more sodium outside the cell, and we have more potassium inside the cell. But how does our body accomplish that? And it's with an antiporter, where things are moving in opposite directions. And the antiporter actually moves free sodium outside the cell and 2 potassium inside the cell. And we expend a tremendous amount of energy all the time to run this antiporter. And if it didn't work, our brain wouldn't work, our heart wouldn't work. the skeletal muscle would not contract. So it's critically important that we can maintain these concentration gradients. And to do it, we have to use energy and use a transporter molecule to accomplish the goal. Okay, so the last thing I always like to review is that idea of osmosis. So, does anybody remember the definition of osmosis? Pardon? What is it? Diffusion through a membrane. Good. Now we have to make that a bit more specific. What's diffusing through a membrane? Water. Yes. Osmosis is the diffusion of water through a membrane. Okay. So what we do is we have to look at two things relative to the cell when we're doing this. Okay. So if we have a cell and we have cytoplasm inside the cell, and all our cells are in an aqueous environment because we have this keratinized stratified squamous that keeps all that water inside of us, then all our cells are in interstitial fluid. And so what we have to do is we have to look at how much stuff is dissolved, we call solutes, over how much solvent there is. And in this instance, the solvent is water. And it's an excellent solvent because it is a polar molecule. All right? So what we would have to do is look at the relative amount of solute to solvent in both compartments. So the word tonicity is a relative measure of that solute. So what we say is hypotonic, hypertonic, and isotonic. So if it's hypotonic, what we would say is we would have less solutes inside the cell and more solutes outside the cell, and the cell is hypertonic. Okay. So that would be an example if our reference is a cell, that we are in a hypo situation. In a hyper situation, we would just reverse that, and we would have more solutes outside the cell than inside the cell, excuse me. I'm just going to put water here now. And then we would have less solutes outside the cell. Well, the interesting thing is that the way students usually get confused with osmosis using hypertonic and hypotonic is that you're focusing on the solutes, the stuff that's dissolved. And if you convert that to focusing on water, then it helps a bunch, okay? So let's say in this situation that we have less solutes here and here. So let's say we have 5% solutes here, and then let's say we have 10% solutes here. Okay? So what we're essentially saying is we have 5 grams in 95 grams of water, and here we have 10 grams in 90 grams of water. So where's water more concentrated? Inside the cell. So what direction is water going to diffuse? Outside the cell. Does that make sense? Okay, and then we would reverse it down here. And water would go the opposite direction. In an isotonic situation, iso means same. So in an isotonic situation, you would have equal solutes in water on both sides. And water moves back and forth, but not in a concentration. So if that makes sense to you, then this will make sense. So if we put a red blood cell in a normal solution, then water would move in both directions, but the cell would not change shape because the cell is not taking on additional water or losing water, right? So now, if I put the cell in a situ. If the cell is hypotonic, then what's going to happen is there's going to be, in the hypotonic solution, there's going to be more water out here. There's going to be less water in here, because the cell now is hypertonic to the solution. So if the solution is hypotonic, the cell is hypertonic. That's the thing you have to understand. And so the cell is going to do what? Take on water. And because we don't have cell walls that prevent the cell from increasing in size, our cells actually this is what the guy was doing who figured out that membranes were by lipid layers. He just took red blood cells, put them in distilled water. That's why when you listen to those medical shows, you've never once heard start a IV of distilled water. So what do they always say? Start a IV of saline? Do they say normal saline? So the word normal implies what? Isotonic. Yeah. So the other thing you do, this is a kind of fun experiment to do, is you can take alcohol, the ethyl alcohol, the kind you drink, and you can put a 10% solution of ethyl alcohol in a beaker and drop red blood cells in it. And then you just put your blood in a hypertonic solution. So now the cell is hypotonic, and all the water is going to vacate the cell, and the cell is going to shrivel up. Ever wonder why high blood levels of alcohol kill you? Because all your neurons are shriveling down. Yeah. So we call that crenation. Now, this picture is so cool, but only if you recognize something. So I'm going to jump ahead, and then we're going to come back to this picture. Okay. So yesterday, what I suggested to you was that we can keep cells that are square, cuboid, columnar, squamous, because cells have an internal skeleton called a cytoskeleton. So this is an example of a cytoskeleton. And it's comprised of microtubules, intermediate filaments, microfilaments. That's stuff you probably learned that the flagella were made of and cilia were made of in general biology. But what happens is all of these fibers inside the cell attach to the membrane in specific locations. And because they're attaching to the membrane, then they can guide the shape of the cell. So what would happen if you have a round cell or a disc cell like a red blood cell, and all of these cytoskeleton components are attaching to the membrane, and you shrivel the cell up? Well, what happens is the membranes collapse around the cytoskeleton. That's why the cell becomes all jagged like that. These are where the points where the cytoskeleton was attached to the membrane. And as the cell is losing water, the membrane is just collapsing around the cytoskeleton. It no longer looks like a disjoint cell. Isn't that cool? Yeah. Not so cool if it happens, but cool to think about. All right. So I don't want to spend a lot of time on this because we're going to review it when we get to where we really need it when we're doing the kidney and stuff. But in physiology, we don't use hypertonic and hypotonic much to describe the relationship between fluids inside your blood versus outside your blood. So if we were comparing interstitial fluid with blood plasma, we really don't use in physiology hypotonic and hypertonic. And what we do is we actually look at the amount of dissolved substances in the water itself. And so what we use is a term called an osmol. And an osmol is how much sodium you can put in the water. And so we refer to it as osmolarity. So what you'll see, what you see oftentimes is a value expressed like this, 300. This is milliosmoles. And that's what we use in physiology instead of hypertonic and hypotonic. We're actually measuring the amount of dissolved substances relative to the solvent and looking at it in terms of a mole. So you remember what a mole was? Little critter with a big toe that digs underground. So it's a given amount of an atom, right? So when we get to like kidney physiology, we'll talk about milliosmoles, and you'll see it expressed in your textbook. So that's all I want you to know right now. It's a way of expressing concentration. All right, so we've been talking about things moving in and out of cells. The other way we can do it is that a cell itself can take something in, and so if you had a bacteria in your body, so a common one you can get from uncooked meat or contaminated meat would be E. coli, little rod-shaped bacteria. And your body is a wonderful environment for E. coli because that's the most common bacteria in your gut. So we keep a body temperature of 37 degrees Celsius. 98.6. We keep that body temperature, and E. coli loves it, and it can replicate itself every 12 minutes at that body temperature. So the other way to look at your body is you're just an incubator for E. coli, which then the E. coli helps us, right? But if it's in your blood, bad news, and it'll replicate itself every 12 minutes. So you can go from a few E. coli you took in from contaminated food to a gob of E. coli that killed you. And that's why people die. All right. So your white blood cells are the ones that are supposed to take care of these. So what do? A big white blood cell recognizes a foreign invader. We'll go later how all that happens. And it just starts engulfing the bacteria. So when the white blood cell engulfs the bacteria, what do we call it? Phagocytosis. Yeah. Yeah. Our white blood cells are specialists at phagocytosis. They engulf those bacteria to destroy them, viruses to destroy them. Okay. So we think of phagocytosis as a cell taking up a solid. All right. So we have these wonderful cells in our body that do many things for us, but sometimes get too big, bigger than we like them. And they're fat cells. And we circulate fat in our blood. But fat is a liquid. And so what happens is when fat droplets come out, these cells love to come up and then transport them into the center of the cell where it will be stored. So since fat is a liquid, then what fat cells specialize at is penocytosis. Yeah. Those are just two examples of where phagocytosis and penocytosis go on in your body. Okay. So the other thing that happens, and so we can actually do a quick review of a bunch of cell stuff, is that you have a nucleus because you have DNA. And a sequence of information on DNA is called a gene, right? So what we do when we need to do something is we have to read a gene. What's the molecule we make from a gene so that our body can, so the cell can use it? Do we make new DNA or do we make RNA? RNA. So what, do you remember what the RNA is called we make? mRNA. Remember, it's the messenger. So it's messenger RNA. And the messenger RNA goes out here, and let's say we're going to do a protein, like a hormone. So what was the organelle out here that makes the protein? Ribosomes. And if it's for export, the ribosomes are associated with roughy R. So now I have my messenger RNA make my hormone, and now I'm making this protein hormone. Now I have another apparatus that packages that hormone for me into little vesicles. So what's that? Golgi apparatus, the packaging center of the cell, right? And then what happens is that little package goes to our cell and is released to the outside of the cell. So that process of packaging and then having that vesicle released to the outside of the cell is called exocytosis. Yeah. So pino, the two P's, pino and phago, are going into the cell. Exocytosis is the way it is out of the cell. All right, yesterday we looked at the stages of mitosis, but I don't want to review that. But what I want to do is I want to kind of review a cell cycle. So when daughter cells are formed from mitosis, they're in interphase. So in interphase, all the DNA is uncoiled so that we can actually read it. Does that make sense? And that's why in interphase, the nucleus doesn't stain dark and we can't see DNA, because it's all uncoiled. All right? So in interphase, to be able to read genes, you have to uncoil your DNA. To prepare the DNA to be duplicated, to prepare the duplicated DNA to divide, we coil it all up. And that's why the chromosomes become obvious in mitosis, because the DNA is all coiled up. All right? So it's like a slinky. Right? So in interphase, we actually divide it into 3 periods of time. So once the daughter cell is formed, the nucleus is in G1, where it duplicates all the organelles, because it just shared everything with its sibling cell. Okay. And cytoplasmic components. So the cell grows and maturates to its adult size, shape, and organelles that it needs to function. That's what happens in G1. All right. Now, as the cell ages, and this is what we're trying to unravel right now, which is going to be revolutionary in medicine, is how the cell cycle genes are controlled, and why a cell knows it's supposed to get ready for another mitotic event. Because once your heart formed and it became adult size, all of the cells in your heart go through G1 and don't do anything else for the rest of your life, and will not divide. That's why heart damage is permanent. So something triggers the cell to know it's supposed to replicate its DNA. So there's a period where the cell does rapid replication of DNA. So that's called S, the S phase of interphase, because we're going to duplicate the DNA. We're going to take the 46 chromosomes that each of us have in a cell, and we're going to convert them from a single DNA molecule to what we call a chromosome with 2 sister chromatids. Now remember, one of the sister chromatids is going to have half of the old DNA and 1 new strand, and the other one's going to have the other half of the old DNA and a new strand. So what we're going to do is duplicate this DNA that's a double helix. Right? And then once we've done the replication of DNA, then the cell expends energy getting ready to divide. So what's the organelle that organizes mitosis? Centrioles, yeah. So you got to make sure you have 2 centrioles. And then you got to make sure you got all the proteins to make spindle apparatus. And so once you've done that, now you're ready to divide, because you have all the equipment you need to do a cell division. And then you would go through mitosis. So the way to think about it is, there are 3 phases of interphase, G1, which is actually gap 1 and gap 2, because we didn't know what the cell was doing. It looked like it was doing nothing, like a gap. Gap 1, synthesis, and gap 2. Those are the phases of interphase. The phases of mitosis are prophase, metaphase, anaphase, telophase. 2 different phases of a cell's life with subunits, okay? All right. Well, because cells divide, then cells can goof up. When cell division goes astray, it's not always a happy event in the body, right? So we have words that describe things that go on based upon mitosis. So a neoplasm, anytime you see the prefix N-E-O, it's new. So what are we making? We're making new plasm, new cells. So a neoplasm is the abnormal formation of tissue. And a good example would be cancer. So cancer is neoplasm, right? What normally happens, recall, is we go from a polygonal cell, which is the vascular division, and in the epithelial tissue we just talked about, we're going to form a squamous cell, a cuboid cell, a columnar cell. So the cell has to differentiate during that G1 phase and reconstruct its internal skeleton, so it changes shape. Right? So when that doesn't happen, then we have words that describe cells that somehow don't differentiate like they're supposed to do. So anaplasia is the loss of differentiation in a cell. So what we would see is that we have a polygonal cell that should have become a squamous cell, but it stayed polygonal. And then it doesn't function well. So anaplasia is that loss of differentiation in cells. Dysplasia is where cells look abnormal under a microscope, but they're not cancer. So women typically, through their reproductive years, have to go in yearly for a pap smear. And what they're doing in a pap smear is they're collecting cells off the surface of the cervix. And they're looking at them under a microscope. And what they're looking for is either anaplasia or dysplasia occurring, where the cells change their shape. So one thing we know is that the human papillomavirus causes cells not to become the normal shape. And by the way, your cervical cells would be squamous cells normally. So they would actually be squamous cells normally. And so what you could get, a report back, is dysplasic cells, and that means that they don't look normal, that the cells are changing the way they look under the scope. If it said anaplasia, then it's like, oh, they're just taking those polygonal cells, that's not good. Yeah. So hyperplasia is just an increase in cell size. So bodybuilders have hyperplasia of skeletal muscle. They're making their muscle cells bigger. Yeah. So one way we would see that is usually as men age, the cells in the prostate begin to swell. So you get hyperplasia of the prostate, and then that narrows the urethra, and then elderly men have trouble peeing. And so then we have these cool drugs like Flomax that are designed to decrease the swelling in the cells so you pee better. Yeah. Right? Met? Pardon? Yeah. You know, what I figured out, and I never worried about it until I was helping take care of my elderly parents and seeing what was going on with them. Then you got to be brave to continue to age. Because my stepfather has hyperplasia prostate, so he's on Flomax. So then when they both have dementia, and we would get their little pills out, label it in their containers, Ray, Martha. And then my mom took my stepfather's pills one morning. She doesn't have a prostate, but she had Flomax. Okay. So metaplasia is a transformation of cells or tissues from normal to abnormal states. That's typically what cancer is the transformation of cells from one state to another. And then apoptosis is programmed cell death. We start making our skin cells, 25 days, and by 25 days later, they're dead and coming off the skin. We make red blood cells. They age and die in 120 days. So we have a lot of programmed cell death in our body. And it works as long as we're doing mitosis right. But when we stop doing mitosis right, this programmed cell death creates all kinds of problems. That's why elderly people's skin gets so thin, is because it's programmed cell death, but they're not doing mitosis like they used to. And so it gets really thin, and almost, in a real elderly person, it's got a really weird feel to it. Yeah, because of, yeah, it's like tissue paper, yeah. It's like, oh, don't touch them, you're going to rip their skin off. Yeah. So, have a wonderful week.